The overall goal of this procedure is to harmlessly and rapidly label the surfaces of living cells or virions with bioactive constructs. This is accomplished by first preparing a function-spacer-lipid or FSL construct solution. Next, an equal part of the construct solution is mixed with an equal part of the solution of cells or virions. As a third step, the mixture is incubated for 60 minutes at 37 degrees Celsius. The final step is to wash the modified cells or codicites or modified virions or codevirions and to use them experimentally as usual. Ultimately, surface-modified live cells or virions can be visualized through all routine experimental analysis techniques, including flow cytometry, microscopy, and agglutination.
The main advantage of this technique over existing methods, like covalent labeling, is that it is rapid, robust, flexible, and does not affect the vitality of the modified cell or virion.
To prepare the FSL construct stock solution, first add 1 ml of diluent to the product file to reconstitute the dry FSL product, creating a 1 mg per milliliter stock solution. Sonicate the vial for 30 seconds. Aliquot the stock into 100 microliter sterile containers and store the containers at 2 to 8 degrees Celsius for up to 1 week. To prepare working FSL construct solutions for insertion, just prior to use, sonicate the solution again for 30 seconds to homogenize any micelles. Then dilute the FSL construct in buffer to the concentration required. Store the working solution at 2 to 8 degrees Celsius for up to a week. After resuspension in lipid-free media or PBS, centrifuge the cells for FSL modification free of unbound lipids. Then, pack the cells in one